Okay. Oh my gosh. I will speak a little softer. I have a loud voice, and they told me I have to use the microphone. So I'm using the microphone. So you might have gathered from my introduction, I'm actually a microbiologist. I'm using Stickleback as a model, and I'll explain that to you in just a minute. But that explains this a little bit, this title. Why would a microbiologist be interested in fish? This was actually a pretty big jump for me that I jumped into during my postdoc. Can I turn this down?
Turn the volume down.
How?
No, you can't do it.
Oh, awesome. Do I have to have this for the recording?
No,
Or
it's
can
not that. It's that when the ceiling blower goes on, go in the back.
Okay. I'm gonna do this for a minute until she figures it out. So my official title, when I usually give a talk, is the influence of host genetic background on the effects of the disturbance of microbiota in host development. That's the official title of what I kind of do. But really, it comes down to a microbiologist trying to adapt to evolution. So the outline of my talk is to really introduce myself and my lab. I have a new lab at the University of Alaska, Anchorage, but I'm pretty proud of it. We're getting some good data already. And then I'm going to talk about the projects that we have. We actually have 5 projects. I'm only going to talk about 3 today. We're interested in microbiota membership, how the host genetic background contributes to. Microbiota membership, the immune response to microbes, and then the recovery from the disturbance of microbiota. And I'll explain that to you in just a minute. But let's start out with my lab and who I am. So I'm a microbiologist. This is driving me nuts. I'm a microbiologist. As an undergraduate, I studied medical microbiology and immunology, so I really studied how microbes interact with the host, but it was always in a pathogenic relationship. So as an undergraduate and as a lab tech for 3 years, I studied regulation of toxin production in Staph aureus, which is a pathogen. And then as a graduate student, I studied unicellular parasites and how they interact with the host. So I studied this cute little parasite called Toxoplasma gondii that forms these cysts, and these cysts are in fun little places like your brain, and when they hatch out of the brain due to a change in your immune system, then they wreak havoc in your brain and other tissues as well. So I studied genes that are responsible for the ability for this little parasite to get from your stomach to your brain. All this is to say, my background really is in pathogenic microbes, but as a when I was finishing up grad school and thinking about a postdoc, I considered, do I want to continue on with this relationship between pathogenic microbes or switch over to beneficial microbes? And I was lucky enough to have Margaret McFulney come to the University of Wisconsin-Madison as a new faculty member, and she really got me interested in host microbe interactions. So as a postdoc, I studied host microbe interactions, and now I have this fun little lab, and I wanted to introduce them before I get too deep into this, who are studying the relationship between beneficial microbes and the host. And in later things, if you ever want to talk to me about this, I'm also interested in coming back to pathogenic microbes. I'm not going to talk about it today, but we are exploring the relationship between beneficial microbes and then preventing pathogens from colonizing. But first I wanted to introduce the students in my lab, the members of my lab, because I'll be talking about their work. These are my undergrads, Coral, Kelly, and Kenny. And then I have a lab tech, Ruth Eisenberg, who is actually at an interview at UConn right now for graduate school. So I'm looking for a new lab tech if anybody wants to come to Alaska. My postdoc is Dr. Emily Luskak, who's been in Stickleback for quite a while. Ryan Lucas also is my master's student, and he has a couple of years of experience in Stickleback. And then Tim Stevenson is really helping us with our microbiota data. So let's get into the meat of the work that we're doing in my lab. What we're really interested in is how does the host genetic background contribute to host microbe interactions? So I keep talking about these beneficial microbes, and they're also known as the microbiota. It turns out that we have complex communities of microbes that are living on us or in us. And the microbial communities are different from one part of the body to the other. I'm really particularly interested in the microbes in the gut. The microbes in the gut are really complex. There are thousands of different species that live in there. They have relationships between themselves. They also can stimulate the host and provide a number of benefits for the host. So for example, they can stimulate the development of certain tissues and cell types, and we can study that in our fish. They can prevent colonization by pathogens. they stimulate the immune system. They can recruit innate immune cells, including neutrophils. And I'm focusing on this because my first initial studies have been in these innate immune cells that are the first responders really to microbes. And their responsibility is really to present microbes to the rest of the immune system. Now, the microbiota in the gut is not only really complex with thousands of different members, but they also vary from individual to individual. So this is a graph, and you're going to see other graphs that are similar to this, in which we use 16S sequencing, not us, but the Human Microbiome Project in this particular case, use 16S sequencing to identify which microbes were in the gut. This is looking at the phylum level. If you remember, King Philip can order 5 golden spoons. This is the highest level. When you get down to genus, this looks like a mess of different colors. But if you look at the phylum level and take a broad look at which microbes are in there, you'll see that in humans, we tend to have firmicutes and bacteroides and some proteobacteria. But the other interesting thing about this, and each color represents a different phylum of bacteria, the other interesting thing about this is that each one of these is a different individual. And you can see that none of them are exactly the same. So there's a lot of variation from one person to another. And that's true for every animal that we've looked at, from humans to mice to fish to koala bears to lizards. So why would we be interested in this relationship between this complex microbial community, especially if they're not exactly the same from one person to another or one individual to another, and the host? One of the reasons for that is because when this microbiota is disturbed, or when our immune response to microbes is disturbed, we have pretty drastic consequences for the host. So for example, I like to focus on irritable bowel syndrome because this is directly related to what I do, because I'm interested in inflammation in the gut. So here we have a patient who has a very colorful patient, a very transparent patient who has irritable bowel syndrome, and he basically just has this really inflamed tissue in the gut. And this is really due to an influx of neutrophils. So here we have, if we take this section, if we take this tissue and we just cut it like this in really thin strips and throw it on a slide and stain it, this is what a healthy gut would look like. It has lots of nice structures. It has some immune cells in there, but it's a manageable amount. But if you take a patient who has ulcerative colitis or any of the other irritable bowel diseases and do a cross-section and stain, you'll see a ton of purple. These are tend to be neutrophils or other innate immune cells. And you can see that the general overall structure of the gut is definitely different than the healthy gut. And this is due to the incredible amount of inflammation. And the other thing that happens when you have a disease like this is that you have a change in the microbiota. Now, this is just taking a really quick look at who's there and averaging. Remember, I said there's a lot of individual differences from one individual to another. But if you take all of those communities and average them out, you get a pretty little pie chart with a couple of different colors. And you can see that the ones for the healthy patients are different than the microbiota for the patients who have inflammatory bowel diseases. And again, each color represents a different phyla of bacteria. So the community structure changes in these diseases as well. Now, the reason I'm really interested in this is because there are also a lot of genetic changes. And I heard that this is a class, so you'll be quizzed, so make sure you write all these genes down. They're very important. They're not very important. They're important for the people who have the diseases. What's important here is that there are many genes that are involved in these inflammatory bowel diseases. If you were going to make a model organism to study this disease, and you wanted to find out the relationship between these different genes and how they contribute to inflammatory bowel disease, you'd be doing an awful lot of crosses, right? You knock out one gene, and then you damage another gene, and then you cross those mice to get another population that has maybe 2 genes, and then this would take an awful lot of crosses to make something that's complex. So if I'm interested in how the host genetic background contributes to the relationship between the inflammatory state of the host and the microbial community, I don't necessarily want to use a host that has a fairly uniform genetic background. I really want a host that has a lot of natural genetic variation. So when I started as a postdoc, I started in this lab that used zebrafish as a model. Turns out zebrafish are really inbred. The lines that we use in the lab tend to be really inbred, but it turns out that there's this really great population of of fish that we can use called Stickleback. I was interested in this because if you go back in the studies of host-microbe interactions, these are images from 1949 from Life magazine, they started out using animals with a lot of natural genetic variation. They used these rabbits and they used these mice that had a lot of genetic variation. In the meantime, in the last 40 or 50 years, we've switched over to these inbred lines that don't have a lot of natural genetic variation. So identifying genes that contribute to the relationship between these hosts and microbes becomes a little more complicated when you're using inbred lines. So I decided to switch over to these little fish called Stickleback. I was in Bill Cresco's lab. Karen got me hooked on host-microbe interactions and the immune response to microbes, and then Bill got me hooked on the evolutionary processes that drive differences in different populations of these fish called Stickleback. I was really lucky to work with Bill because he used Alaskan populations. It just so happens I'm from Alaska, so you know, going back home was a really a great idea for me. This is my home. I'm from Kotzebue. I live down here in Anchorage, and it turns out that there are hundreds of lakes around Anchorage within driving distance that have different populations of Stickleback in them. And you can think of these as little genetic experiments that have been going on in nature in Alaska for sometimes as short as 40 or 50 years, and other times for hundreds of thousands of years. So we have these different populations of Stickleback that have been evolving in isolation from each other, and sometimes there's some movement between the bodies of water, so you get some mixtures, you can follow some of that genetic variation. Other times you just have these populations that have been evolving in isolation, and they're evolving different relationships. My hypothesis is that they're evolving different relationships with their microbes. And one of the reasons I think that is because these different lakes have different phenotypes themselves. They have differences in pH, they have differences in nitrogen levels, they have differences in water, differences in where their water is coming from, and all those and other things are going to influence the microbial community that's there. So it turns out that Stickleback, when they evolve in isolation from each other, you may be very familiar with this from the work that Mike Bella's done here, they also evolve different phenotypes. And this is just a stain looking at the bones, and bones of, see, now I'm going to get nervous because I am not an evolutionary biologist, and Mike Bella's sitting here, and he's going to point out later all the things that I've gotten wrong in this particular section. But you can see that fish from one population, this is an Anadromous or Oceanic population, has an awful lot of lateral plates, and a really quick, simple phenotype to look for is a decrease in the lateral plates in these freshwater populations. I'm oversimplifying it, and I'm mixing up the words Anadromous and Oceanic, and Mike will correct me on that later. But in addition to having these wonderful natural genetic variation experiments that have been going on all over Alaska, we have other reasons to use fish. So it turns out, oh, that's nice. This is a drawing of a fish that you can't see anymore. It turns out that fish have a lot of benefits compared to other organisms that we might use, like mice. We get really large clutches from a single cross, so we cross one male and one female, and we can get anywhere from 80 to 200 eggs. The juvenile are optically transparent, so here's fish that's developing in an egg, and you can actually watch the eggs divide under the microscope, which is fascinating. If you ever get to work in a fish lab, you should, because it's amazing to watch. And then you can watch them develop all the way to the juvenile stage. They're still transparent. But a cool thing about that is that we can also label different cells in these fish then, and watch those cells move around in live fish. And we can fluorescently label microbes and watch them move around in live fish, which is a lot of fun. I'm not going to present on that data today, but that's something that we've done. There's also evolution, and the evolution and development is thoroughly described for Stickleback. I'm just using this as an example that not only is it well documented and well described, but we've also been able to, not we, this is Kingsley's lab, has been able to show that there are different processes that occur in Stickleback that are conserved all the way to humans. So, you know, I go home and I tell my parents I'm doing important things, and they say, why in the heck are you using a fish? It's for these reasons, and the fact that if we find something really interesting in fish, we may be able to identify whether or not that also occurs in humans. In this particular case, Kingsley found a gene that was responsible for the difference in this plate armor, and Mike is definitely going to tell me I got this wrong. And then what they found is that this enhancer, this also affects the ability of the length of the human foot and is responsible for the chimp foot evolving into a human foot. That's oversimplification. Again, not an evolutionary biologist. So there are many shared pathways. And in addition to this nice one that we can observe very easily, there are also many shared pathways involved in host microbe interactions in the innate immune response with mammals. My former postdoc advisor, Karen Gilliman, has shown an awful lot of that. And of course, I've talked a lot about natural genetic variation. It turns out we also share a lot of gut microbes. So if we just take a really broad look at the microbes that are in the gut, and we compare humans to stickleback, each one of these blue bars indicates that it's present in that particular species, and we share an awful lot of blue bars between humans and stickleback. So there are a lot of microbes that are shared between humans and stickleback. And importantly, there are microbes that are found in stickleback that are not found in some of the other, that are found in stickleback in humans that aren't found in some of the commonly used animal models. And then this is where I'm really going to flub things up. And now I'm really worried because I see Krishna in the background, and he's going to tell me I got this wrong. We can also look at genetic distances. So this is comparing the genetic background of one host to another. And we can look at it on this FST scale, where 0 means that they are basically the same species. And then 1 means that these genetic differences are so broad and so different. There are so many different genetic differences that we can say that this is one species versus another. And just an example that most people are going to be familiar with, we can show that chimps and humans tend to have a value somewhere closer to 1. Chimps versus bonobos, which are much more closely related, are going to have something closer to 0.5. And then chimp species A versus species B is going to be a really small number, somewhere around 0.2. So we're interested in whether stickleback can be used as a model for human diseases, and we wanted to determine whether their natural genetic variation reflected what we see in human populations. So we focused in on this different speciation right down there in this FST little chart that we have. And very big thank you to Krishna for correcting on, or for helping me put this together an hour and a half ago. You can see that if we look at non-African versus African human populations, we get a value somewhere between 0.1 and 0.2. And then if we look at human populations that are more closely related, like European and Asians, those population differences are closer to 0.1. In stickleback populations, if you take one oceanic population and compare it to another, we get a value that's really close to 0, indicating that they're very closely related. In the populations that we study, these differences between oceanic and freshwater, these oceanic, some of them are purely oceanic in that they live in the ocean. Most of them are anadromous in that they swim back and forth between the ocean and the freshwater, depending on the part of their life cycle that they're in, but we're going to call them oceanic. You can see that their values are pretty close to this European and Asian. And then if we also look at compare freshwater populations against each other, so a freshwater population that's been isolated in this lake versus a freshwater population isolated in this lake versus this lake, we can see that those mix in here somewhere around oceanic and freshwater. So the genetic differences in our populations are somewhat similar to what you would see between, say, European and Asians. Oh, good, I spelled something wrong on a slide. So hopefully at this point I've convinced you that stickleback are a good model for host microbe interactions. Because I'm interested in identifying the relationships between, or characterizing these relationships between microbes and their hosts, I really wanted to look at the microbial communities. And so what I'm going to do now is just explain to you some of the tools I have for looking at these relationships. So I can raise these fish in the presence of complex microbial communities. For those of you who are not microbiologists, this is not drawn to scale. These microbes are not this big. It's fine. They're very small. But the conventional fish have a really complex microbial community. I can then take those fish and I can raise them germ-free. I just You saw that they first develop in early stages in these chorions, these eggs. I can sterilize the surface of those using some bleach and iodine. I'll show you that in a minute, and then raise them in the absence of microbes. So that gives me an idea of this very drastic change, raising the fish with microbes and raising the fish without microbes. I can also add back individual microbes. So here we had a complex microbial community with a whole bunch of different types. Now we can mono-associate, just adding one particular type of microbe back to this relationship and find out how that one microbe interacts with the host. And then we can add back 2, and we can add back 5, and we can add back little communities of 20 or 100 different microbes and really play with which microbes are there. The next thing we can do is we can start thinking about, that's the microbial part. We can manipulate the host, and we can collect these fish from different populations, so we can kind of manipulate the genetic background that way. We can also look at the output. So here we're using a neutrophil stain to identify the neutrophils in the gut. So what we've done here is we've taken a big old fish like this, and we cut them in really small slices like this, and then we stain them with a stain that stains the neutrophils, and then we just go through and count these neutrophils. And I say just, but this takes like a long time. So this is very labor intensive. We're finding other assays now to quantify our immune response. But the data that we're going to show you today was collected like this. So we have this neutrophil stain to look at the immune response. We can isolate the microbes from the gut. So we take this fish, open him up, grab its gut, and plate the microbes on these plates. And then we can take these individual microbes and we can freeze them down in a freezer and pull them out whenever we want to play with them. We can label them with fluorescent molecules so that we can follow where they go in a live host and see how they interact with each other. We can do DNA and RNA sequencing. Hopefully you're all very familiar with that. I'm not going to talk about the transcriptome analysis that we've done, but we've started playing with transcriptome analysis. And then we can isolate and sequence the DNA from both the fish and from the microbes to find out who's there. We can also look at morphological differences. So we can take our fish. This is a juvenile fish. I switched it on you. We had an adult fish here. This is a juvenile fish. And we can take different morphological measurements. So standard length, swim bladder area and size, eye diameter in many different ways. And we're looking for other morphological measurements that we can do. But these are the ones that we started out with initially because they're easy to see on these fish that we take pictures of. And then I'm going to tell you about some behavioral data in which we put our fish on a grid like this. And then it's hard to see these little guys, but there are fish in these little red circles. And we can follow, we can videotape them video them for a while, and then count how many squares they go into and how much time they spend on the perimeter. And we have all these behavioral assays that we can do to look at how the microbes are affecting their behavior as well. Okay, whole lot of introduction. Hopefully I've convinced you at this point. This is an amazing system, and we're going to find out amazing things. So what amazing things are we finding out? First, we're going to start out with microbial membership. Right now, I'm focusing on Alaskan populations. When I was a postdoc, I was actually in Oregon. So we started out with the populations we had. We had Alaskan populations in the lab. I'll show you some data on that. We also had Oregon populations that were wild. So we took a couple of wild Oregon populations, one that's oceanic. It's actually anadromous, but we're going to call it oceanic, and then a freshwater sample as well. This is the first time I ever did field studies. Those of you who are stuck in a lab, you should find a model system where you can do field studies. They're much more fun. So then we took these wild fish and we dissected out the guts and we isolated the DNA and we did some magical sequencing and some analysis. And this should look really familiar because you saw a similar chart on one of my introductory slides. Each color represents a different order, we're in order here, a different order of microbe that's present in this particular individual, and this individual is different than this, is different than this. This is the wild oceanic Oregon population. And we saw, okay, we have individual to individual variation, which we expected, so yay, good for us, we can do some sequencing. And then we looked at our freshwater population, and wow, there's a whole bunch of differences there. This was really interesting. What we discovered is that there weren't so many differences on which microbes were present at the order level. There's some speciation differences. But what's really cool is that the freshwater population had a much more diverse microbial community than the oceanic population. There are lots of caveats to this particular study. We need more numbers. We need to be able to control for the environment. There are a lot of things that we need to do, but this was fun. We found that a wild Oregon freshwater population had more diverse microbiota than the wild oceanic population. We wanted to compare those to the lab-raised fish, and I wanted to build a database of microbial isolates, and so we took out those guts and we plated them, and then we compared the microbes from the plating between the oceanic wild Oregon fish to the freshwater wild Oregon fish to the oceanic Alaskan lab-raised fish to the freshwater Alaskan lab-raised fish. Each dark color represents a microbial community colony type that was highly abundant in that particular community of population of fish, and each white represents those that were absent. These microbial types were absent from this particular population, and then the colors in between are the gradient of the different types of microbes that were present. One of the things that we found right off the bat is that the wild population had microbes that the freshwater didn't, so that was interesting to us. We hadn't compared them before. We also found that there were some microbes that were present only in a freshwater or an oceanic population, but not present in the other. So that was fun as well. We had some sort of speciation, population differences going on there. And then we also found that although these wild fish were raised in the wild, they shared a lot of microbial types with those raised in the lab. So this was also fun because then we could show that the lab-raised fish were not completely different from the wild fish. In doing this, I made a collection. I've added to that with the populations that I'm looking at in Alaska. I now have over 280 microbial isolates. They're from 4 different phyla, 22 different genera, a whole bunch of unidentified, which are going to be fun to figure out what they are. Fish ranging from juveniles all the way to adults, Oregon and Alaska fish, wild microbes, and lab-raised microbes. So these are all microbial isolates that I have in a freezer that I take out and play with on the benchtop. So our findings. There are differences between wild oceanic and freshwater gut microbiota. The wild oceanic and freshwater stickleback differ in alpha diversity. That was the difference in the microbial types that were in the diversity of microbes from one population to another. And the lab-raised stickleback share many microbial community members with the wild populations. Now, these were just initial studies. This is really a baseline for figuring out what kind of things that we want to focus on in the future. One of the things that we're focusing on now is comparing the microbial community in the water versus the microbial community that's in the fish. We're doing this a number of different ways. One of the things that we're doing is we're just going out to these wild populations that we can drive to in Alaska, grabbing some water samples, grabbing some fish samples, comparing the water chemistry and the microbial composition of the wild populations in the water, and then comparing those to the microbial populations in the fish. And we're trying to figure out if different populations of fish select for or against different microbes that are in the environment. So that's fun. Where are we collecting from? As I said, we're located here in Anchorage, so we've collected from a bunch of lakes up in Talkeetna, in part because it was a pretty drive, but we had some biological reasons too. You can ask me about those later. And then we have a bunch of populations that are right around Anchorage. Here's the city of Anchorage. We collect from Westchester Lagoon. We collect from Cheney Lake. We're doing a longitudinal study to see how the microbiota changes over the course of a year in wild populations. And then some fish from Big Lake and Mud Lake that I'll tell you about in just a minute. So these different freshwater fish from different areas, and some anadromous fish as well. And then we're going to be expanding on these collections in the spring. And for those of you who are stuck in the lab, I just wanted to show you what working in a field in Alaska looks like. It's pretty amazing. And I have a bunch of lab work that goes on in the lab as well. Again, I was a bench scientist for 15 years. It's really fun for me to do field work now. So we do have some initial studies. We went out, we collected these fish, we started looking at the microbes that are in the gut. This is just some sequencing analysis showing that, hey, we can sequence in our lab. It wasn't a fluke. It's not just something I could do as a postdoc. It's something I can do as an assistant professor. So here we have our wild-caught populations. This is looking at the phylum level. So this is a higher order than the, looking at a different level of the characterization of the microbes than we did before. Before we were looking at order, now we're looking at phylum. Individuals vary from one individual to another. Their microbial communities are, seem to be much more variable in our Alaskan populations in general, in our adults, than they were in our Oregon populations. We have proteobacteria, we have firmicutes just like humans do. We have those bacteroides just like humans do. We also looked at some juveniles that we raised in the lab. What's really fun is that we actually colonized these lab-raised fish with microbes from our wild fish because we wanted to have a microbial community that was as similar as possible to our wild population. And sure enough, we were able to do that. So that was really exciting. There wasn't much of a difference between our adults and our juveniles, and that's just what we're showing here, that we can have the same microbial community in our wild versus our lab-raised populations. So that was exciting. Because we're interested in how the host genetic background contributes to this relationship, we're taking some fish, we're exposing them, these fish from different populations, we're raising them in the lab. One of the problems with our wild experiments is that these fish are in these wild waterways that have different microbial communities. If we bring different populations and raise them in the lab together, we can get a better idea of what the host genetic background is doing in selecting and for and against the microbial communities. So we're taking a couple of oceanic populations and a couple of freshwater populations, putting them in nets so that they're exposed to the same microbes that are in the water, and raising them together in these community garden experiments. And these are just getting underway. So that's the microbial membership. I'm also interested in the host immune response. As you can imagine, these microbes, these fish have been evolving in these bodies of water that have different microbial communities. They have different nitrogen and carbon levels. They have different pHs, et cetera. Our hypothesis is that fish that are evolving in these different microbial communities will also evolve a different immune response to microbes. So to measure this immune response to microbes, we take our fish in their juvenile stages. We actually take them somewhere between 2 and 48 hours. We make them germ-free, so we sterilize the surface using this pretty easy protocol where we do some soaks. And then we raise them up to 14 days post-fertilization. And that's because at 14 days post-fertilization, we have to start feeding the fish. And we're working on a germ-free food source right now. We inoculate at about 10 days post-fertilization. The gut for Stickleback opens between 10 and 14 days post-fertilization, typically around 12 days post-fertilization. And then in the work that I'm going to show you now, we did that neutrophil stain. We raised these fish germ-free or conventional in the lab, and then we stained them and sectioned them and looked at their number of neutrophils in the gut. When we did this for oceanic fish that were raised in complex microbial communities, that's what the CV stands for, that's that conventional fish. When we raise them in the presence of lots of different microbes, we have a lot of neutrophils in the gut. So this is fun. We can count neutrophils. When we raise our oceanic population in germ-free fish, in the absence of microbes, we see very few neutrophils in the gut. So this is interesting. In fish that are raised in the presence of many microbes, we have a lot of neutrophils in the gut. And in our germ-free fish, we don't have very many neutrophils. What happens if we start adding back individual microbes? We get some rescue with the one population of or with the one microbial type that we used, we get some rescue of the number of neutrophils in the gut, indicating that this particular microbe isn't responsible for all the neutrophil recruitment, but is responsible for some of it. I've actually done a bunch of these now with many different types of microbes, and so now I have a better idea of which microbes are actually stimulating the immune response in oceanic fish. This is oceanic fish. I'm interested in how the host genetic background contributes to this relationship, and so we compared our oceanic fish to our freshwater fish, and lo and behold, there was not a difference in the number of neutrophils in the gut, whether we raised them conventional, germ-free, or monoassociated, indicating that in this particular population of freshwater fish, there was not a drastic immune response to microbes in our freshwater fish, but there was a drastic immune response in our oceanic fish. This is a paper we published last year. So this is fun. Now we have an assay that takes a really long time that shows that there is a difference in two different populations between their immune response to microbes in the fish. So now what we're doing is we are, here's a little conclusion to that. So now what we're doing is we're going through and finding other populations and starting to look at their immune response to microbes as well. And we're hoping to find out if there are freshwater populations that have different immune responses than this first freshwater population that we looked at. And those are in really early stages right now. Whew. So that's the microbiota membership. That's the immune response to microbes. So now we're starting to play with this idea of actually manipulating the microbiota and figuring out how that affects fish from different populations. And our idea is that fish, like humans, are not all affected by microbial disturbances in the same way. So it turns out in some really preliminary studies in kids that some kids respond to antibiotic exposure by having a hyperinflamed or becoming more susceptible to pathogens, or antibiotic exposure can have really drastic effects on their immune system in the gut and their overall health. And other kids can be exposed to antibiotics and not be quite as disrupted. Their development is not quite as disrupted. So we're interested in figuring out if this happens in our little fish, and if we can explain some of this differences based on natural genetic variation, based on their genetic background. So this is just to show you that we can do many gross morphological measurements, which I discussed earlier. So what we did is we first looked at a really drastic difference in the really drastic changes in the microbial membership, which is to raise fish germ-free versus conventional. And in that initial study that we did with just one group of fish that was raised germ-free and conventional, we saw that in that one population, there was not a difference in standard length, there was not a difference in eye diameter, there was not a difference in head depth, there was a difference in the swim bladder area and swim bladder size. And so that was fun because now we have this morphological measurement that we can do in these early fish and see that there's some sort of difference. Now we're going to repeat this for other populations. So that was the drastic difference of germ-free and conventional. We also saw that there were differences in the exploratory behavior. So again, we take this flask of fish and our little tiny fish, and we put a grid underneath them, and then we just videotape them and watch where they go. We look at the total number of squares that the fish explore. We look at the novel squares, so do they go from, and how many new squares do they go into, and how much time do they spend on the perimeter of that flask. In our oceanic population, this rabbit slough population, we saw that principal component 1, when we throw all this data together in our data analysis, principal component 1 explains or differentiates conventional fish from our germ-free fish. And it turns out that this is based on the differences in how often the fish spend how much time they spend on the perimeter versus traveling around, or going to the center of that flask. And so what we found is that our germ-free and conventional fish are significantly different in our oceanic fish based on principal component 1. This is driven by differences in the parameter squares, and the germ-free fish spend more time on the parameter than our conventional fish. So here we have an indication that in our oceanic fish that our germ-free fish have a different behavior than our freshwater fish. We have one population of freshwater fish that we looked at, and we can see that principal component 2 separates our germ-free from our conventional fish for our freshwater population. And what's interesting is that it's not the same behavior. It's actually that the conventional fish are swimming greater differences. So we have this difference in these two populations in this extreme condition of germ-free versus conventional, in which the oceanic fish are spending a different amount of time on the perimeter, but the freshwater fish are traveling different distances based on whether they're raised in the presence or absence of microbes. So this was fun. And our conclusion, our overall conclusion, is that there's a gene by environment influence on both oceanic and freshwater behavior, but they're driven by different behaviors. So this is fun. I mentioned that a bunch of times because I really like this stuff. I really do think it's fun. Okay, that's really drastic. Conventional versus germ-free. That's a really drastic change in the microbiota. What happens if we just tweak that microbiota a little bit? So here we're starting to look at antibiotics. Dr. Lessig has an NSF grant to study how chronic exposure to antibiotics affects different populations of fish. And the idea here is that there are many different antibiotics that are actually washed away. When we take them, they go into the environment in our different bodies of water, and these fish are evolving in the presence of these antibiotics. And these antibiotics are changing the microbiota of the water, but they're also changing possibly the evolution of the fish, because we're drastically changing the microbiota. So she's going to look at long-term chronic exposure to low levels of antibiotics to mimic levels in wild fish habitats. That's her NSF grant. And then our master's student, Ryan, has an NIH-sponsored INRI grant looking at short-term exposure to clinically relevant levels of antibiotics. So this is mimicking what kids are exposed to when they're young and they get an ear infection and they go to the doctor, and the doctor gives them too many antibiotics and that disrupts their microbiota. We're going to explore whether or not that disruption to the microbiota early in development is affecting fish from different populations, hosts from different populations in different ways. We've already done some initial studies. So we just started out with tetracycline. We exposed fish from different populations, 2 freshwater populations, 1 oceanic populations. We exposed them to no antibiotics, some ethanol because that's what our drug is dissolved in, low concentration of tetracycline, which is Dr. Leskak's experiments, and then high tetracycline, and that's for Ryan's, our master students' experiments. This is just showing where these different populations come from. For those of you who are interested, again, Anchorage is right here. So they're populations that we can just drive out to and grab some eggs in the spring and play with them. This is just a timeline of what we do. Our fish are raised in the lab. At 12 days, we add some antibiotics. Again, that's when their gut is just starting to open. This is looking really early. We just wanted to do a quick experiment to see if there was a difference. So we terminate at 14 days post-fertilization, and then we look at a whole bunch of different stuff. Our initial studies, I'm going to go through these kind of quick because we're running out of time. This is the eye diameter. So we have our freshwater population, an oceanic population that's going to pop up here, and our second freshwater population that's going to pop up here. This is our control, ethanol, low concentration of tetracycline, high concentration of tetracycline. In our first freshwater population, there is a difference, there's no difference. There's a small difference between our antibiotic exposed and our control fish. In our oceanic fish, again, this is really early, there's no difference at all. In our second freshwater fish population, there's a big difference between the tetracycline treated and the non-treated fish, indicating that there's a contribution of the host genetic background to this relationship between disturbance of the microbiota and development. So this is for eye diameter. We see similar results when we look at standard length and when we look at swim bladder. There's no difference in behavior. So this is just looking at Big Lake, one of the freshwater populations, low TET, high TET, ethanol and control, and there's no difference in their behavior. And that was done by a student that was just working with us for a summer. So she did a lot of work in one summer. There's Haley. Okay, so overall, on antibiotic disruption of microbiota, we found that there is a gene by environment interaction that appears to drive the response to antibiotic exposure. In freshwater, we have increased developmental instability, which is much more dramatic in one freshwater population than another. Or oceanic, there's not a difference, and there's no influence on behavior. That was fun. It was a short study. We're going to repeat it. We are also setting up a long-term exposure, so that was only 14 days, a juvenile stage of the fish. We're going to take this out to a year. The typical lifespan of a stickleback is 2 years. We're taking it out to a year and taking samples when their gut opens, before and after the onset of adaptive immunity, and at the very end when the fish are sexually mature. And then looking at the same things that we were looking at before. Okay, so host genetic contribution. Our big conclusions thus far: host genetic background contributes to host microbe interactions. Does it contribute to microbial membership? Probably, but we need more information. Does it does the host genetic background contribute to the immune response to microbes? Yes, but we'd like more information. And does it contribute to the recovery from disturbance of the microbiota? But yes, but we need more information, so I still need funding, so just keep that in mind. And I need more students, and I need a new lab tech. So if anybody wants to go to Alaska for a couple of years, come talk to me. I've been very lucky to recruit some really talented people to my lab. I've only been there for a year and a half, but we've gotten a lot of stuff done. And so these are the people in my lab. I work with Brandon Briggs and Janelle. We work together on looking at the microbes in the water. He's a specialist in microbes in the water. Frank Von Hippel is at UEA. Now he's at NAU. We're collaborating on some fun projects that I didn't get to talk about. These are the people in the Gillerman and the Cresco labs that helped me learn about fish because I knew nothing. Poking does our histology, and I've been very lucky in a couple of short years to have lots of funding. So with that, I will take any remaining questions. Not all at once, people. I can't handle it. Yeah.
Okay, so I'm not an immunologist or a fish person.
Okay.
But I am interested. I mean, one of the things that's been talked about for a long time in the geochemistry community is the fact that fish guts could be a different kind of environment where you could get low oxygen or something. So do you have any sense about what actually is? I don't think people have hypothesized that methane production occurs in guts. No one has really, if you make the samples anoxic, they produce methane.
Yeah.
But is anything known about what's going on in these things in the field, or how these different organism classes, do you find any diagnostic organisms that might relate to that?
Yeah. So that goes in two different directions. One is Project 7 and 8 that I didn't talk about. We are interested in gut morphology. So it turns out zebrafish, a lot of the early work in host microbe interactions in fish occurred in zebrafish. Well, zebrafish don't have a real stomach, and their gut is just one small tube that comes off of a big intestinal bulb into a tube. In stickleback, we actually have a true stomach, and then we have an anterior and a posterior gut. So my hypothesis is that, driven by nothing, is that the microbes in the gut are actually probably going to be in a more anaerobic environment than they would be in a zebrafish gut, in part because we have those closures, and that helps close off microbial communities, which helps them consume some oxygen and then make this anaerobic environment. I'd really like to figure that out. I'm working with Jonathan down the hall, who specializes in tiny probes. to figure out if we can figure out the pH and the oxygen levels in the gut. Have we found any microbes that would be indicative? We have found archaea, which I am super excited about because I've never studied archaea, and why not go into a new field when I'm just 1.5 years into my assistant professorship and I don't have tenure? It sounds like fun. Archaea are really great at producing or using methane and producing methane. And so this, it's only one small study and it's only by 16S, but wouldn't it be fun if we found archaea in stickleback guts and then could start exploring if those guys are actually producing methane and is that changing the environment of the stickleback gut. Archaea are in our guts, and they're very abundant in cows that produce a lot of methane, but they're also found in our gut as well. So that would be another fun area to explore. What's allowing these archaea to survive in stickleback guts, and is it the same environment that's allowing them to grow in human guts?
So when you make these germ-free fish,
yeah.
Presumably the guts are not germ-free also.
No, they're all germ-free. So the beauty, yeah.
But are you sterilizing the food? Are you sterilizing the water?
Yeah. So the beauty of fish is that they actually grow in these chorions, and the chorions are resistant to chemicals. So I can throw bleach and do an iodine soak on these stickleback for a long time, and that kills the microbes on the surface. And our initial studies by in situ hybridization, 16S sequencing, and 16S PCR indicates that the environment inside the egg is sterile. So if we sterilize the surface of the egg and raise these fish in germ-free water, then we're raising fish that are germ-free. They don't have any microbes anywhere on them. Right now we can only do it to 14 days post-fertilization in stickleback. As a postdoc, I was working on making germ-free food for zebrafish, and we're going to use some of those techniques for stickleback as well. Yes?
On the anatomy still, so
yeah.
Do these fish have the equivalent of M cells in higher vertebrates or not? Or if not, how they sense and sample about some microbes?
Yep. So we know that stickleback have macrophages. Pretty sure. We know that they have neutrophils. We know a lot more about zebrafish immunity than we do about stickleback immunity. We know a lot about the transcripts that are present, indicating that there are the immune cells that would be similar to humans. We're figuring out the stains to prove that they're there, and some of my undergrads are working on some of those projects. What we do know is that in zebrafish, the gut architecture is different than humans in that, in humans we have these deep crits, and you have stem cells that form in these crits, and epithelial cells come out of the crits, and at the top of the, on the villi of the intestine in humans, we have, epithelial cells that started there and came up here, and that we have this great blood flow underneath, or in these villi that allow immune cells to go in and sample the outside. And then we have those great pyre patches with a whole bunch of immune cells. We don't know that for sure for stickleback. I've done some initial analysis looking at mucin-producing cells, and I found that they definitely have mucin. They have the goblet cells in the gut. We know that they have neutrophils in the gut. I've stained them. I've looked at them. We are working on looking at some of the other immune cells that are in the gut. In part, we're doing this through RT-PCR, just looking for transcripts again. And we have a nice transcriptome paper that was just accepted. So that Clay at the University of Oregon led the transcriptomic analysis on, and he's found quite a number of immune cell genes that are present in the gut. So there's probably a great immune cell presence there. Now we need to characterize them. Yes.
I'm just curious, did you have a reason for looking at swim bladder? You found significant results in swim bladder. So that's important for almost all teleos fish. So
yeah.
I was curious, is there a reason that you think you're getting an effect there?
So I'm interested in it for two reasons. One, it's really easy to measure, and I have undergrads, so I want them to be able to get some quick measurements. So that was one reason. Another reason is that the swim bladder, some people have used the swim bladder as kind of an analogy to lung function in humans, in that there's this great air interface between the body and the the swim bladder, the organ. As far as I know, there are no actual microbes in there. So my thinking, there's no microbes. So my thinking right now is that microbes are stimulating the cells at the epithelial layer, and those have a downstream effect on different parts of development. We know that the swim bladder in zebrafish has this really cool bifurcation and developmental markers that we can follow during development. In zebrafish, we're just starting, probably Schwarp knows all about that, but we're starting to characterize in our little populations the development of the swim bladder. The swim bladder is fun because it has a nice little cell layer that has a whole lot of oxygen, and then it has cell layers around that have slightly less oxygen and that come into contact with other stimuli from different parts of the body. And so, long story short, I don't know yet, but this is something that we're eager to start playing with and learning more about. Yeah, I started rambling there, and I realized, oh, can't do that.
I have another question?
Yeah.
So the effect of antibiotic on growth, is it, you think, similar to what we have in terrestrial animals, that the industry been using antibiotics and have growth in chickens, cows,
yeah,
whatever, from an energy standpoint, that the immune system has cost energy, so if you don't use it for that, you think we have similar situation here?
So we're just starting to explore that. We know that we see similar effects in Stickleback as we do in our in mammals that get antibiotic exposure, and that our fish that are exposed to antibiotics are bigger than our fish that are not exposed to antibiotics, indicating that it could be that. I'm really interested in that immune response, so I can't wait to start identifying and characterizing the immune cells that are there. That's my hypothesis right now, is that we're going to see similar pathways, and that we'll be able to use the differences between the populations to determine if there are immune cells, obviously, that are different, and immune responses and transcripts that are different, but then also starting to think about metabolism and thinking about or metabolites and thinking about other microbial signals, that because you know the microbes are making all these really great small molecules that can travel distances in the body outside of the gut, and so we're interested in following those as well and determining if those are also affecting the development of the host in different ways because of different genes. Last one.
Do your germ-free fish grow or develop at a different rate than non-germ-free fish?
Yeah, so we, in a paper that we published 2 years ago, we actually looked at lots of different measurements of germ-free fish. We looked at standard length, we looked at gut development because that was a particular interest of ours. We looked at goblet cells. Those are all the same between our germ-free and our conventional fish. The thing that was different was the immune response. That's the short version of the long story. But yeah, the short version of the long story is that there's not a huge difference between our germ-free and our conventional fish in development, in those two populations that we looked at. Okay.
So I think it's time to get some pizza. There is more time to discuss with her over there. Also because most of the day yesterday, everything yesterday has been cancelled. We have been discussing how to make more time to meet with Kat. So tonight we are going to Club Red. I'm taking Kat to Club Red at 4:30. So we can have a nice dinner and also discuss more with her. And after Club Red, we are also, Mike is offering for people interested to actually go to his place and have more informal discussions on that fascinating topic. So, do you want to give your advice?
It's really easy if you don't screw up. It's 2 Maple Avenue. It's in Stony Brook. And note that it's about 100 meters from the Stony Brook train station, because there's also a 2 Maple Avenue in every town in this area, and I get lots of people's mail. So when you put it into your phone, 2 Maple Avenue, Stony Brook, and if it doesn't come out near the train station, you're going to the wrong place. And let's say, what time? 7 o'clock, if you'd like to come on, 7:30, and you know, do your best to park. You can walk. You can easily walk to my house.
You can easily park on the street.
No, with snow.
Oh, not with the snow.
But you can do it.